Hello everyone. First of all, I would like to thanks to the organizer for providing me the opportunity to present my work as a video presentation in the 5th International Eurasian Conference. It's me, Sayyida Saima Razak. I'm a PhD scholar of Dr. Zafar Edzadi Center for Proteomics, University of Karachi, Pakistan. My topic of presentation is cardiomyogenic differentiation of human umbilical cord-derived mesenchymal stem cells by overexpression of cardiac transcription factors. This is the overview of my presentation. We all know that cardiovascular disease are a group of disorders of heart and blood vessels. As prevalence ratio of cardiovascular disease is globally high because of some common risk factors like smoking, high blood pressure, high cholesterol, obesity, physical inactivity, and diabetes. According to an estimation, worldwide mortality ratio of cardiovascular disease is 32%, where this ratio in Pakistan is 29%. There are a number of cardiovascular diseases that prevail, but the most common are congenital heart disease, coronary heart disease, cerebrovascular disease, and heart failure, which is also called myocardial infarction. Myocardial infarction basically it's a condition where blood flow stops or reduces in any part of heart, and as a result of this disturbance, it makes the heart tissue become impaired or non-functional. Currently, number of treatment strategies are available like thrombosis, stent implantation, bypass grafting, and transplantation. But none of these can repair the damaged tissues of heart and repair the normal functionality of heart, with the exception of heart transplantation. But the ratio of heart transplantation is quite limited due to insufficiency of donor, and it is very difficult to control the immunological reaction when the heart transplantation performed in the recipient body. So in medicinal sciences, cellular therapy has been introduced because of its unique characteristics, and in cellular therapy, stem cells are widely used for the transplantation purpose. Stem cells are unspecialized cells having multi-differentiation and regenerative potential. There are various sources of stem cells because of different classification, but on the basis of their sources, stem cells are classified into two types, which is embryonic stem cells and adult stem cells. Embryonic stem cells are the stem cells which are obtained from embryonic sources, and they are pluripotent type, means these stem cells have capability to differentiate into all types of cells, whereas adult stem cells are the stem cells which are obtained from adult sources, and they can differentiate into specific cellular lineage. One of the example of adult stem cell is mesenchymal stem cells, and mesenchymal stem cells can easily be isolated from various sources like bone marrow, adipose tissues, skin, muscles, placenta, umbilical cord, and its blood. Human umbilical cord is considered an enriched source of mesenchymal stem cells because of less ethical concern, and it can easily be isolated because it is considered a medical waste. Moreover, it facilitates the allogenic and heterogenic transplantation, and they can differentiate into specific lineage by various growth factors, small molecules, and transcription factors. Now, what is transcription factors? Transcription factors are the DNA binding proteins because they have DNA binding domains which facilitate the expression of a specific sequence of DNA. There are number of cardiac transcription factors in the literature have been reported which involve the differentiation of stem cells into cardiac lineage, such as GATA binding protein 4 and myosin enhancer factor is one of them. So, the aim of this study is to assess the role of cardiac transcription factors for the differentiation of human umbilical cord-derived mesenchymal stem cells into cardiac lineage. This is the methodology of the research which was focused for targeting the research objectives. So before starting the experimental work, IRB approval was taken from the Ethical Committee of University of Karachi. Then umbilical cord tissue was collected from the obstetrical unit of Dow Hospital. In this study, healthy women having the C-sections with the age range of 20 to 42 were included, and make sure these women did not have any medical complications like high blood pressure, diabetes, and any other viral or bacterial infections. Then umbilical cord tissue was collected from the patient after taking their consent agreement. After collecting the umbilical cord tissue, cells were isolated and allowed to propagate. Then characterization of stem cells were performed by using the standard criteria. For this, MSCs specific markers were detected by using different techniques like immunocytochemistry, flow cytometry, and adipogenic and osteogenic potential of MSCs were also detected. After confirming that isolated cells were mesenchymal stem cells, their transfection was performed by using the two cardiac transcription factor, which is GATA binding protein 4 and myocyte enhancer factor 2C. Then efficiency of transfection was detected by cardiac gene expression and protein expression analysis. There are 4 experimental groups are included in this study. Group 1 is control where no transfection was performed. Group 2 having the cells which was transfected with GATA4 transcription factor. Group 3 having the cells which was transfected with MAF2C transcription factor, and Group 4 having the cells where co-transfection was performed. Towards the results and discussion. This is the pictorial representation of isolation and morphology of human umbilical cord-derived mesenchymal stem cells. After isolation and propagation of umbilical cord-derived mesenchymal stem cells, their characterization was performed according to the standard prescribed criteria. In this figure, A section was represented where MSC-specific markers were detected by immunocytochemistry, whereas in the B section, MSC-specific markers were detected by flow cytometry, and in the C sections of this figure, adipogenic and osteogenic potential of mesenchymal stem cells were detected. Confirming that isolated cells are mesenchymal stem cells, the transfection was performed, then efficiency of transfection was detected by the gene expression analysis. In this figure, you can see that in each transfected group, statistical significant expression of cardiac transcription factors were detected in their respective group. Here, morphological changes was shown in each transfected group. In this figure, A section represents the normal cell where no transfection was performed, whereas the B section represents the GATA transfected cells, C section represents the MAF2C transfected cells, and the D section represents the co-transfection. Cardiac gene expression analysis was performed. For this, early and late stage markers of cardiomyocytes were detected. In this figure, you can see the significant expression of early and late stage markers of cardiomyocytes were expressed in each transfected group. After cardiac gene expression analysis, cardiac protein expression analysis was also performed by immunocytochemistry. Here, both early and late stage markers of cardiomyocytes were detected like connexin 43, cardiac troponin T, GATA4, and NKX2. This is the quantified results of protein expression analysis. In this figure, you can see the early stage markers of cardiomyocyte, which are connexin 43, GATA4, and NKX2.5, were significantly expressed in all of the transfected group, especially in the co-transfected group. Whereas late stage marker of cardiomyocyte, which is cardiac troponin T, were not significantly expressed in all of the transfected group. Study is that the cardiac transcription factors have potential to differentiate umbilical cord-derived mesenchymal stem cells into cardiac lineage. The expression of early and late cardiac genes were significantly high in all of the transfected groups. Co-transfection has synergistic effects on cardiac differentiation. So this research highlights the regenerative potential of genetically modified cellular therapy, and it could be a promising strategy for the regeneration of myocardial injury. This is the published article of the. At the end, I would like to acknowledge this work with my supervisor, Dr. Kamal Hanif, with my co-supervisor, Dr. Irfan Khan, and hEC for providing the indigenous scholarship. Then all of the faculty of my institute, Dr. Zafar Zaidi Center for Proteomics, and all my collaborators and my friends and colleagues. Thank you.